Welcome to the lesson on the control of the cell cycle. As you are aware, most of a cell's life is spent in a stage known as interphase. During interphase, a cell is going to perform its special function. It's going to do its special job. So if this is a red blood cell, it's going to be ferrying oxygen. If it's an immune cell, it's going to be fighting disease. In addition to doing its special job, a cell is going to grow during interphase, it's going to copy its DNA, which is very important, and it's going to get ready to divide. If you look at this microscope slide of an onion root tip, you can see that most of the cells here are in interphase as well. So here's a cell in interphase, here and here and here, and you can tell it's interphase because you don't see the chromosomes yet, and you can see this dark spot in the nucleus called the nucleolus. There are certain cells in our bodies that never leave interphase. So once they mature, once they grow to their adult size, they get stuck in a phase known as G0 and they don't divide anymore. So we have to take care of them throughout our entire lives. That is part of the reason why spinal cord and brain injuries are so severe, because those cells in particular, nerve cells, enter this G0 phase and you can't make copies of the cells because they don't divide. Now, cells in our bodies and in living things only divide when conditions are favorable and they receive signals to do so. So how does this all work? How does a cell know when to divide and when not to divide? And that's what we're going to tackle next. There are certain genes inside of our bodies and they are called checkpoint genes, and they produce substances like proteins that cause the cell cycle either to advance, to move forward, or to pause. And these checkpoint genes monitor different things. So for example, they monitor DNA copying. Has the DNA been copied during the S phase of interphase, or is it not complete? Is there DNA damage? If the DNA is copied, is it copied well? Are there mutations in that DNA? If there's mutations, the cell doesn't want to go ahead and divide. It wants to fix those mistakes. Is there enough nutrients available? Because the cell isn't going to reproduce. It's not going to produce more cells if those cells are just going to starve because there's not enough food or water in the environment. And then cells also respond to these things known as growth factors that are telling them this is the time to divide. So for example, if you get a cut on your hand, growth factors would be released that would stimulate those skin cells to divide and reproduce and fill that gap that you caused by when you sliced up your finger and got a paper cut. So again, this is interphase over here, if you remember. And an interphase, this part of the cell cycle is interphase, and it's broken up into three parts. G1, which is the first gap phase during which the cell grows, and then S phase, which is when the cell is copying all of its DNA, it's duplicating its chromosomes, and then G2 phase of interphase, where the cell is growing some more and it's getting ready for cell division. And then this tiny slice here is the mitotic phase, and this is when the cell is actually actively dividing. So there are different checkpoint genes that make sure that at every single step of the way, things are being done properly and it makes sense for the cell to divide. So there are these things known as checkpoints, and you want to think of checkpoints like it's an inspection station where the cell is going to assess and make sure everything is okay before it goes to the next stage of the cell cycle. The first checkpoint is going to be right here in the G1 phase. So during G1, the cell is just a baby cell and it's growing and it's getting bigger. And then it hits this first checkpoint, the G1 checkpoint. And the checkpoint is represented here by this traffic light. And here, the cell is going to check a few things. It's going to check, is the cell big enough? Is it healthy enough? Are environmental conditions good? If everything checks out, the cell is going to move past G1 and it's going to enter S phase and it's going to start copying all of its DNA. And then it's going to go to G2, and all of a sudden in G2, it's going to hit that second checkpoint, the G2 checkpoint. Here, the cell is going to be checking the DNA. Is all of the DNA copied? Did we do a good job? Did we copy everything? It's going to also check, how is that DNA? Is it filled with mistakes? Are there mutations in it? Because mutations can be very bad. Mutations can lead to cancer. So at this point, the cell is going to make sure that any mistakes that are present get fixed before the cell gets the okay to go forward and enter the mitotic phase, which is cell division. If everything checks out, the cell is going to go to mitosis. In mitosis, there's a checkpoint, and that checkpoint is in metaphase. So the cell is going to do prophase, and then it's going to go to metaphase. And in metaphase, there's a checkpoint to make sure that all of the chromosomes are lined up, single file, in a straight line, perfectly in the middle. It's going to make sure none of the chromosomes are off to the side or kind of not exactly where they should be. This mitosis checkpoint is going to ensure everything looks like this. If the chromosomes are properly lined up, then the cell is going to go through the remaining stages, anaphase, telophase, and then it's going to go to cytokinesis, and you're going to end up with 2 healthy daughter cells. Cells also respond to density and coming into contact with other cells. So most of the time, if there's plenty of nutrients and space available, the cell can divide very rapidly. So if we take these cells and put them into this petri dish with plenty of food and space, they're each going to divide until they run into each other and they fill up the entire space. As soon as they hit an edge or they hit each other, they stop dividing. So at this point, the cells will not divide anymore. If you go in there and you wipe out half, or the middle part of these cells of this petri dish, then the cells that are towards the middle are going to start reproducing, and they're going to fill that gap until they run into each other, and then they're going to stop reproducing again. So these checkpoint stops, running out of space and stopping cell division, these are things that normal cells do. Now, cancer cells are cells that don't do these things. Cancer cells ignore the checkpoints. So you want to think of these checkpoints as it's a traffic stop, right? The police officer's pulling you over, checking your license and registration before it lets you go forward. Cancer cells don't stop. They don't care if the police officer stops them. They don't care if there's a traffic light. They just go through and they keep reproducing and reproducing and reproducing, and they make these really, really bad copies of themselves. They don't respond to signals. And the other thing that cancer cells ignore is they don't care if they run into another healthy cell. They don't care if they run into any type of boundary. They don't care. They're just going to keep reproducing, and they form these things known as tumors. And tumors are just these bad types of cells that are not normal, they're irregular, they look irregular, they don't function properly, and they can cause a lot of damage. Cancer can spread to other tissues through the circulatory system as well. And cancer divides even without anything telling it to divide, even without any type of growth factors. There are 2 types of tumors that can form. The first type is less severe. They're known as benign tumors, and they remain at an original site. They're kind of self-contained. Malignant tumors are worse because they're tumors that spread to other locations by something known as metastasis. They basically travel through your circulatory system, and they can spread to different parts of the body. So, other things about cancer. Characteristics of cancer cells: they lost the ability to stop dividing. They're out of control. They're immortal. So our regular cells only divide a certain number of times, and they end up dying, and they're replaced by other cells. Cancer cells are basically cells that they don't die. They just keep surviving. They're not subject to contact inhibition, which is what we saw with the petri dish. So they don't care if they run into another cell or an edge. They just keep dividing. They can metastasize, so they can spread into other tissues, and they are unspecialized. So these are all characteristics of cancer cells. Now, there are treatments out there for cancer, and there are certain treatments that are good for localized cancer, so cancers that are staying in one place, like radiation and surgery. During surgery, a tumor can be removed, and they'll typically remove not just the tumor, but some of the healthy cells around it, just to make sure we got everything. And then during radiation, they're using high doses of radiation to kill cancer cells and help shrink tumors. So you want to think of things like x-rays. Like when you get an x-ray, that's a very low dose of radiation. For cancer treatments, you get a much higher but very targeted dose of radiation. And then there's other ones that treat the system, like chemotherapy is a class of drugs that attack rapidly dividing cells. And cancer is one of those types of cells, so it'll attack cancer cells. The problem with chemotherapy is that it attacks other rapidly dividing cells, like the cells that make hair. It can attack sperm cells as well. So any type of cells that reproduce very quickly, chemotherapy will attack. So it can be very hard on the body. And then there's biological therapies, and these therapies work with your body's immune system and try to get it to recognize cancer, to fight cancer, to target it better. And there's a lot of research going on with biological therapies to target specific types of cancers. And the consequences or the side effects of biological therapies are often much less severe than, let's say, chemotherapy. So all of this is pretty negative, and cancer is really an awful disease, and it sounds pretty depressing. But there's actually good news out there. So every time people donate money to help cancer research and they support cancer research, they are putting their money to good work because it's working, and we are coming up with better treatments. And just in the last decades, the death rate from all cancers has declined 27%. That's a huge amount. So people are surviving. We have better methods of early detection where we catch cancer earlier before it's able to do a lot of damage. We have much better treatments. People are doing things that are healthier. They're avoiding cigarette smoking. They're avoiding eating things that are really bad for them, that have carcinogens in them. And there's been declines in the death rates of some of the most common cancers. So look at these statistics. There's a 48% decline for lung cancer in men, a 23% decline for lung cancer in women, a 40% decline for death from breast cancer in women, 51% for prostate cancer in men, and then 53% for colorectal cancer. So these cancer rates or death rates from these cancers have been declining. We've been able to save a lot of people. Here's another one showing you some statistics. So the good news is there are a lot of people now who are beating cancer. Just in 2014, 14.5 million people beat cancer. And that number is expected to grow. So by 2024, we think that number is going to be closer to 19 million. So these people who are able to fight cancer due to early detection and due to better treatment. So that is definitely good news. Childhood cancer rates are down, or deaths from childhood cancers are down. And then Americans are kicking the habit, right? There's fewer people who are smoking today than ever. And then science is also coming up with newer and better treatments, right? We are coming up with targeted therapies that are attacking cancer cells and not causing as much damage as chemotherapy. So there is definitely good news, and we are definitely trying to fight this and figure it out. So thank you so much for listening and watching.